All right, welcome everybody. Open Source Antibiotics Series 2 meeting on Friday, September the 11th. And let me share screen so that we know what it is we're looking at. Hopefully that is clear and everyone can see the screen. Okay, so thanks for coming along. The big news, this week is that we had some compounds tested. So I guess the main thing I want to do is to have a quick chat about future design. Just before we get to that, I'll just highlight a couple of things. So we've been having a few conversations offline with various people about what they're doing in the project. Main one is that Lee Graves at the University of North Carolina, Chapel Hill, who had agreed to do the mechanism of action experiments. We had a chat with him and resolved what needs to be done, and that's going to go ahead, I think, next week. Caro Wells, who has the original compounds, has the two compounds that are needed there, which I guess we're trying to be consistent about, the actives and inactives that we're using for the project to make sure that when we publish it, we've kind of, got data on the same compounds, which makes a simpler story. So there's an active and an inactive, and Lee is going to use those two in these mechanism of action experiments, which are talked about in issue 15. So that's good, and I guess we just chase it in a couple of weeks to see how it's going, to see what we get.
So Matt, how long have those compounds been stored?
Oh, well, Alvaro may know that.
I will say less than 2 years, 1 year and some months.
They're solid.
1 year and a half, yeah. But there are, and they were also solid of each one. So I don't know if the compound that they are using is the new prepared compound, or it was a solution that was there. But maximum 1 year and a half. And I'm sure that Caro prepared a new batch of compound.
Yeah, I think it's important to, that if you're going to do experiments like this, you do it on really nice, clean, pure compound, not on stored solutions or anything.
Yeah, it's a good one. I'll chase Caro on that just to be sure. If they're solids, maybe we could check the NMR. I think the lab that's providing the MRSA is nearby to Lee. So they could have a quick test of the compounds versus the organism.
Yeah.
Yeah, okay, good reminder. Thank you, important. I guess I was pleased, by the way, to see that these two compounds still were active and inactive in the latest results that we just got. So there's a confirmation there, which is good. Yeah. Nothing needed on the clearance assays, which are happening in Monash. Hopefully we'll get this back pretty soon. And on some of these other things, there have been some conversations between me and Haifa, the company, and between me and Jim Callahan at GSK. And I just can't quite report yet about those agreements, but certainly interesting conversations which are going places about trying to isolate some metabolites and trying to learn what we can about what was done in GSK on this series back in the day. So I will update as soon as I'm able to say things, and I've just got to clear it with them about what I can say, but there's progress being made at least. And just on point 7, they're still trying to track down the original report from COAD on these compounds. I'm guessing that Bill has that. So we got the report about how the compounds were tested, but not the specific data on these compounds. And one of the reasons why I'm interested is because I guess I kind of assumed that the original hit was found by COAD, but that all of the compounds which were screened, 25 or so compounds, had been screened elsewhere. But I guess we're thinking that maybe they were all screened by COAD, in which case there's a big document there that we need to track down, which has the original data in there. I'm also assuming that document contains the information that we have about their toxicity or not versus, mammalian cells, which I'll come to in a second. But we need that because we need to be sure. All right. So the main thing really is the data from the latest round of screening. So Paul Stapleton, my colleague here at School of Pharmacy, screened these 9 compounds, which Dana and Ed had made. And this is, some of these are repeats. Some of them are to be compared against the known SAR, which we originally had, which is on the wiki page. So there's, some of them are repeats, some of them are new. So I guess there's 2 sets of data here, which we need to combine together. So the original active was this guy, 822, and the original inactive was this guy, 820, which is a very small change, but makes a big difference. And then several of these other ones were synthesized to explore various aspects of things. This compound, this 812, is the one that was, so this is kind of the base compound, which everything is a derivative of, but we realized that we didn't actually have any data on this compound. So we've remade it and found that it in itself is also inactive.
Yep.
And then these are the other new compounds, including the one that Ed made with the phenyl there, which obliterates the activity. So that's an important data point, obviously. And then there are some other ones here that have been made, which are all inactive, apart from these with a sniff of activity when we have the sulfur there. Just quickly on the point that was made by Ben overnight about toxicity, which is an interesting point. So he was saying that he is worried that the chelation possibility between the red and the green here as a metal chelator would make these compounds generically toxic. And Chris, you made the point that, well, why doesn't that happen? Why then are not all these compounds active? Which is a good point. And I just dug up the relevant bit of text from I need to refresh this, from the grant proposal, the original grant proposal, which I need, maybe Dana could check at some point that if this is on the wiki, because I couldn't find it, but I found it in the original grant proposal, which says this. So this is the preliminary data we used to get the grant going in the first place. So all 15 of these compounds that were shown, I think, in the wiki were evaluated for cytotox. All demonstrated similar cytotoxicity in 293 with a concentration of device. So within twofold of 12 micrograms per mil for all compounds. Concentration at 10% hemolytic activity in human red blood cells was greater than 32 micrograms per mil. So there's a window of therapeutic activity. So there is some toxicity, but there's a window of therapeutic activity. But again, it would be nice to see, to check whether that correlates with the activity against MRSA in the first place. I guess that's what we need to see if there's a correlation there.
Yeah, definitely.
And that's why we need that original report. So yeah, so Ben has also said a couple of other things here about the need perhaps to do an analog sweep of the green ring. Now he was originally suggesting that we go from a 2-pyridyl to 3 and 4, and that has already been done. So we know from the original data that the 3 and the 4 isomers are inactive. So you need the 2. But as far as I can tell, those are the only analogs we have. We haven't done anything else at that position, as far as I know, in the green position, apart from now trying the phenyl. So I think that's the limit. And Ben was suggesting other heterocycles there, like I think he said 3 pyrazole or 4 imidazole. I think is what he was saying. So he was suggesting to try to mess around with that position a little bit. And I guess it depends if we can generate those compounds. So he was saying replacing 2-pyridyl with other things was a way of getting their clearance down, which is another one of the things we're trying to do in this project. So maybe that increases the need for us to look at that ring system when we are doing our analoguing. On the bright side, yeah.
Sorry, Matt. I guess the testing that Paul did was the same that the COAT guys, or
we haven't done a proper comparison.
Okay. Well, the results in the most active compounds is almost the same. It's 4 and the other was 2. But I don't know if they are doing something similar or it's a different testing.
Yeah, we got that COAD report right about the same time that Paul was starting his assay. I mean, literally the same kind of hour. So we weren't able to change it, but we need to look at the incubation time, for example, and check whether it's...
Okay, yes.
Well, wouldn't the bead pull down, the protein target searching, if this thing was metal binding, wouldn't it cause a bunch of protein targets to pop up on the screen, so we would kind of know that way if this thing was metal binding?
I don't know. I think seeing a correlation with generic toxicity would be a bad thing. I mean, I think that would be a roadblock right there, right? I mean, we don't really...
Yeah, we don't want a metal binder, but I was wondering if the...
Well, unless it's relevant to the mechanism and allows us, again, a distinction between the pathogen and a human cell. I mean, Chris, you made the point about the fact that if it's a metal binder, it's not necessarily the end of the world, as long as it's selective.
That's right. If the metal happens to be in the active site of your target protein, then that's fine.
Right. So I think, keeping a heteroatom there may be a wise thing to do, given what we found with the 3 and the 4 pyridines being inactive. Maybe that's what we need to do. But I think this raises enough of an issue that we need to keep an eye on the tox screening. Just parenthetically about that, getting very close now to be able to do some of this tox screening quickly here in the School of Pharmacy. Just one last little paperwork thing to get sorted out, and then we may be able to do that here quickly. So maybe we'll have the answer more quickly than we think. But I'm hoping it's in the COAD report, because they should do that by default. Also, I mean, just taking a step back here, looking at any compounds with activity. So these guys with, of course, our original hit and the sulfurs up here, and from the original SAR, the compounds that have activity, here again with the sulfur, here again with the sulfur. I guess we have some oxygens down here, but the compounds with the activity appear to have sulfurs, and I don't know what to make of that, or whether that is remotely significant. But maybe this is something to do, again, with one of the main thrusts of the project, which is to try and work out what's happening to these compounds in terms of their clearance, and whether or not a metabolite is actually the active compound. I think it's too early to say, but there's a little bit of a trend there that our sulfur-containing compounds are the most potent.
Do you get much metabolism in your assay?
Don't know. Who are you asking?
Well, anybody who knows, I guess. No, it's just that if it's an in vitro assay, I wouldn't anticipate much metabolism.
Right. Yeah. Mm-hmm. That's a fair point. I don't know. I don't know about the nature of the assay. That's a good one for Paul.
We could make one of them. I mean, you could make one of the metabolites pretty easily. That OSA 824, the parathioether, there's the sulfoxide and sulfone of that boronic acid available.
Uh-huh.
Yeah.
So you could probably get the 822, yeah, the Mercimic, the one with the 4 microgram molar. 4 microgram milliliter rather. You can get the sulfone of that boronic acid.
Yeah, I mean, I think Ed will talk about this in a minute, because he's going after some of the oxidized compounds. One of the aims of the interaction we're trying to generate with the company Haifa is to show that we're getting oxidation and to try to fish out some of those oxidized species. I mean, that's the whole point. So again, I guess, so some of the data from Monash may also show that there could be a correlation between propensity to be oxidized and potency. I guess that's also something we're trying to look for. But it's a good point. I'll check in with Paul Stapleton about the nature of the assay. Okay, I mean, I might at this point. So one of the other things that that Ben says is, which in this post, which was very useful, is that they do have some compounds that may be relevant to us, and I guess I need to follow up with him about this. some information about molecules that may that they've made on a related project that they may be able to share with us. So that's something I'll be chasing up with him. Always nice to be able to use compounds that have been made already. Mm-hmm. All right. Ed, are you okay to talk a little bit about chemistry ongoing and maybe a little bit about design on the basis of these data?
Yeah.
All right, cool. Let me stop sharing.
Okay, yeah, so I'll just go over Dana's stuff as well.
Thanks.
So she's been working on these kind of couplings. So she's tried to use some different Suzuki conditions with this oxazole, which wasn't working with the initial conditions, but it seems that it's just giving debrominated material. And then she's also been working on these imidazole compounds and trying to do Suzukis on them. So for this triddle one, she's getting decomposition, and strangely for this methyl imidazole, she managed to get this dimer. Not really sure how, but I guess there's a transfer of the boronate. I don't know. But she was thinking of switching the coupling partners, so having like this as the boronate and that as the bromine instead, and seeing if that helps. So yeah, we've purchased that bromopyridine, and we'll see if that works.
Wait, the bottom right dimer, you've not isomerized your ring, right? You've just got two imidazoles coupled. Is that what you mean?
Yeah. So yeah, I saw the NMRs, and it's just, you know, you get 3 singlets, and it's symmetrical, and the mass is doubled.
Okay, because your southwest imidazole is not an imidazole.
Oh, I think that might just be drawn wrong.
That would be amazing if that's happening.
Yeah.
Okay, fine.
Yeah, I think that's just drawn wrong.
Can I just see the paper title now? Facile cyclic hydrazine synthesis with no hydrazine involved.
You never know. All right, great. Okay, I mean, obviously these compounds, because we need to try and make them, obviously these are based on the same kind of core, right?
Yeah.
Two pyridine, which is fine because we should finish what we start.
And then I guess just on my stuff, so I was trying to make the oxidized sulfur compounds. And from last week, I tried the direct oxidation, but it looks like it's given me the N-oxide instead. So yeah, that doesn't seem to work directly. I've put that on hold. I was hoping that maybe Dana's Suzuki conditions would have worked, because I've got the sulfone boronic ester, which I could couple, but yeah, I'm not too sure about that at the moment. Maybe going to microwaves might help. But yeah, and in the meantime, I've also been making more of the core. So I've been scaling up progressively each time. And so for this last time, I've kind of deliberately extracted the aqueous, even though it's like a lot of DMF. But that seems to have like improved the yield almost double. You just have to deal with getting rid of all that DMF, which can be a bit of a hassle, but I guess getting more is worth it. And then, yeah, as Ben was suggesting, so these kind of pyrazole and imidazole compounds. So I had a quick look at the respective starting materials. So I guess we would need like this alpha bromo ketone of these. and yeah, they're not really that easy to get to. So I don't know, maybe there are some alternative isomers of this that are easier, but
I'd be a little
yeah,
concerned about stability too.
Yeah.
The implication from Ben's message is that they've made compounds like this. So you might want to reach out to him and say, do you have any synthetic stuff you could share with us?
Yeah.
They may have a good route. But it could be, in theory, you could imagine, well, it depends. Any aromatic heterocycle with something in that 2 position might work. Maybe it doesn't have to be nitrogen. Yeah. I guess think broadly here. Yeah, okay.
Yeah, I mean, I only had a quick look since this morning.
Okay. I mean, if you could search on that, the core structure, the imidazole with the ring, or maybe with a carbon on the nitrogen, and then an aromatic ring in the northwest, and then just a carbon in the southwest. So find out who's put things in that position and what's available. That would be neat. In terms of doing an analog sweep on the southwest, but maintaining the imidazole for now and maintaining there being something carbon based on one of the nitrogen. So try and keep it fuzzy. Yeah. And I mean, at the moment, keeping the benzothiophene in the northwest seems like a sensible thing to do based on potency, but not ultimately if it's a liability. We don't know it's a liability yet, but at least we're getting potent compounds.
I guess the other compound that would be interesting to make is the replace the pyridine with a pyrimidine.
Yep. I had a feeling that was done. I had a feeling we mentioned that compound before, but that's absolutely right. Yeah.
I believe the one that was mentioned was the one that was from acetylpyrazine, which is a perfume ingredient. It was the one I believe that was mentioned.
Yeah. Okay. Yeah, so 6-membered rings too, Ed, I guess, include that in the search to see what's available. Is there, so the route on the bottom there to get to the core system, do we have any, are there any obvious options if we didn't want to do it that way?
And is the problem there that you're saying this is a low yield, but is there still more in the aqueous phase?
It's pretty messy, to be honest. Like after the column itself is pretty stained. I'm sure probably Alvaro has also encountered this when he made them, but yeah.
Well, you can also try to use another solvent and change DMF for, I don't know, dioxane or THF. But I don't know if it can dissolve the 2 species of the reaction. Also because both are salts, and also the sodium bicarbonate or carbonate. And you can try in a small scale and probably dioxane. I don't know. I think it is reported to use DMF because it can dissolve the pyridine.
Okay.
Really good. But I don't know if the problem is the DMF. Could be better to try other solvent.
I mean, we could look for another polar solvent that's aprotic.
I mean, is the problem you're getting reaction with your pyridine with the acyl bromide?
I would have thought so.
So can you just, if you can get that into solution, can you just add it to a stirred solution of your other reagents? Just add it over an hour or so.
Yeah, we could try that.
Yeah. So I guess I was thinking about an alternative disconnection where you make the alkyl imidazole with something in that position, and then at the next step you couple the ring with a Suzuki or something. Is that an alternative? Because then we could play more easily with that ring system.
Yeah, I mean, I guess that's kind of what Dana was looking at with, yeah, these ones.
Right. So we're having an issue with that, obviously.
Yeah.
Funny. Yeah, that's funny that that's complicated.
You said that the column was stained afterwards. To me, that almost makes me think of like oxidation products. Is this done in vacuo, the bringing of the acyl bromide and the amine together?
I mean, yeah, it's just, you know, under room temperature, 80-degree conditions. It's nothing sealed. Yeah, it's just to air.
That N oxide, the 4792, obviously keep it, prove it, and we'll screen it.
Definitely, yeah.
It's not bad.
Yeah, there are a couple N oxide approved drugs.
All right, good. Okay, I mean, yeah, if we are going to explore that, then I mean, I guess we're doing all the right chemistry already, so that's good. And if we could find a modular way of introducing that ring at the end, that would obviously be the best thing. Yeah. Okay. All right. Great. Thanks, Ed. For I think the only other thing that we were going to mention. I think that's probably everything that we were gonna do. Oh yeah, I mean the one last. I guess Dana's on the train or something at the moment. There was this issue to do with just the impurity that we saw, or the signal that we saw in the NMR last time, that Dana was seeing in some of these NMRs. That was a mobile hydrogen bonding type signal. I don't know if Dana, if you're able to comment on that, whether we resolved what that was. I think Dana's traveling, so it may be difficult for her to talk.
Yeah, so from what I know, we've not resolved it yet.
Okay.
So we were thinking about, I don't know, either me giving that Suzuki a go and seeing if it shows up again, or if she ran the NMR using the bottle of chloroform that I've been using, to see if that's where it's coming from. But yeah, that'll have to be next week.
Okay. I mean, if those samples, I don't know if those samples have a trend of activity, or if they're inactive and we're surprised, or if they're active. It was just a few of those samples, right? It wasn't everything. Is that right? Just got to be a little bit concerned about,
I think she said in some of them. I don't know.
I mean, if some of them have got this and some of them don't, and we're testing for activity, we just got to be sure about what it is we have. The most obvious thing would be a chelated H, but then you've got a salt, which is fine, but we should know if it's a salt from the mass spec. Actually, we wouldn't if it was protonated. I guess it's difficult to work this out, isn't it? It'd be nice if we crystallize it, obviously, if that's doable. But good to resolve before we conclude everything about the potency of those molecules, right? Because we don't want to be making bad conclusions. Yeah, okay. So it's still, I'm going to leave it there because I think it's still important that we resolve what it is. Taking one of the samples, crystallizing it, getting the structure would be the nicest way of showing that we're happy with what it is.
Yeah.
Okay, great. Was there anything else anyone wanted to say?
Hi, Matt, this is Ben.
Hi.
I've just managed to dial in about 3 minutes ago. Sorry. Because I saw there were a couple of comments about the comment that I left at random this morning. So I just wanted to know if there was anything that I can help with on that side.
Yeah, absolutely great. Thank you for coming. Yeah, there were. We were going to chat to you about, I guess, offline about the molecules that you might be able to dig up from the related project if some of those are structurally similar. There was, though, this very, I'll just go to the relevant thing here. So you were saying, if you can see the screen, about the analog sweep of the green ring here, which is a great idea. We've already got data on the 4 and 5 isomers, sorry, the 3 and 4 isomers, which are inactive. So that's been done already, but we don't have much more. So we haven't played with that position. And Ed was just talking about the synthesis with maybe other coordinating heterocycles in that position. And we were hoping that maybe you'd already made some of these things and had a decent way of making those compounds.
So yeah, so just a heads up. So can you just go back to the slide you were just showing with the 3 and 4 pyridar?
Oh, sorry. Yeah.
Right. See the compound in the bottom, not the compound very bottom left, but the next one across, 814.
Yeah.
Okay. So all of our compounds would have been, almost all of our compounds of that type, usually with substituents on the phenyl on the 6-membered ring, you know, methyls and chloros and a bunch of methoxys and things like that. We've also got a couple of examples of the next one over, which is the saturated version, 5,6. We don't have any examples of 5,5, you know, semi-saturated like which seems to be where you're coming from. And the other thing is that most of ours are at the 3 position. So where you have your methylene dioxy phenyl ring in your hit compound, we have an amino linker. So there's a nitrogen between the aromatic ring and the heterocycle, or an oxygen. So it's a couple of steps away, but we've made a lot of these sorts of compounds, and there are a few that may be a little bit closer to the sorts of things you want to test. That's why what I was offering is to go through that set and maybe pull, you know, 10 representative diverse examples out and send them on to you.
That would be amazing.
I mean, some of them we have to ring fence because they're tied up in, you know, they're encumbered because of where they've come from. But I'm pretty certain that at least half of them we're free to do whatever we want with. And have samples. So yeah, because I saw an example that had been put forward. I think, correct me if I'm wrong, but I think Anthony had put forward a version where instead of a phenyl ring at that position, it was a morpholine. We definitely have that compound. And it was interesting, but it was just a heads up because, you know, we were always wondering whether what we were seeing was purely due to sort of metal sequestering toxicity. Because it was still never entirely resolved, because what we would see is that, you know, some compounds we would make would be exquisitely potent against the parasite and have no impact on the background host cell line, and then other compounds with a very small change, all of a sudden, you know, the toxicity on the cell line was the same as it was against the parasite. It was a bit weird. But the things I would point out that you should probably check, I would take, you know, the exact compound you've got here. What is that? 0868. And swap out the pyridyl, the 2-pyridyl. I would put in a 3-pyrazole with and without the methyl group on the nitrogen of the pyrazole, because with the NH, you should see a massive improvement in your metabolic stability, because that pyridyl, I think, is metabolically labile in this sort of system, maybe. And then the other thing to check is that you can get rid of the pyridyl and replace it with an electron-rich phenyl in our series. So, for example, a dimethoxyphenyl or a para-dimethylaminoaniline, which, okay, yes, it's an aniline, but from an SAR perspective could be interesting. And it was those sorts of things, the way you make the phenyl ring a little bit electron-rich, that seemed to also work.
Interesting. Just to clarify on this guy here, 814. This compound has in the northwest this blue portion.
Yeah, and I think I made a comment on that in the comment I put in this morning. I couldn't see the potency of that compound in the first instance, so I didn't know what I was comparing it with. Whether when you went to that 5,6 fully aromatic system, whether you lost potency or not, it was difficult to tell.
No,
I assume you do because that's your hit, but I don't know.
No, this is one of those things. We didn't have it for that, so we've made it and tested it, and it's inactive.
Ah, okay.
So I wouldn't necessarily read anything into that ring being a negative.
Yeah. And of course, you know, the thing I should point out there is that ring, particularly when you have an aromatic ring at the 2 position, I mean, that's a massively privileged structure. There are launch drugs with that, like zolpidem. A lot of the sort of Z drugs for insomnia have exactly that core. So there's a lot of stuff out there if that's active. So yeah, I'll see what I can pull together that is unencumbered. How much do you need to test?
Ed, what was it for Paul? MIG or something?
Yeah, MIG.
Shipped as a solid, is that okay?
Ideally.
Yeah.
And where would I have to ship it to?
Here.
UCL?
Yeah.
Yeah, okay, it's easy, no problem. Okay, I'll see what I can do in the next week or so.
Amazing. That would be really good. Yeah, thank you. I
And
mean, yeah,
how many-
representative set would be very helpful.
Yeah, and what we can do, I'm going to suggest 10 or so in the first instance, just to get a diversity scan across our SAR. And then if we see hits, we can obviously dive deeper.
Yeah, that's amazing. And if there was chemistry done by somebody to vary that ring on the imidazole, if there was a good disconnection there, or a good approach for that, that would be great to know, because Ed and Dana would like to look at that.
Okay, so when you're making compounds like 814, that's made through a one-part multi-component reaction where you condense an aminopyridine, an aldehyde, and a tosmic reagent, an isonitrile, sorry, not an isonitrile, an isocyanide. Yeah, an isonitrile, same thing. Yeah, so the chemistry is a little bit capricious because the isocyanide can be sometimes be so reactive you can't do anything with it, and other times it can be so stable that it doesn't react. But generally, you know, it's really easy to scan around the green pyridyl ring because that's where the aldehyde comes from. So it's that comes from an aldehyde. The ring, the 6-membered ring of the 5,6 can be substituted easily because it's amino pyridines. And then the bit off the 3 position in the northwest, as you said, that comes from your isonitriles.
Okay, that's good to know. Do you have that anywhere?
That chemistry?
Yeah.
Yeah, totally. So we're actually still working on that series for one of the 2 parasites, and that's being done by Laurie Farrens.
Oh, really?
Who you know. So they're making compounds like that. We made a ton of these compounds at Wuxi. Yeah.
That makes it easier. That makes it easier. Okay, so we
the stuff that Laurie's doing, that's under an NIH grant, in which it's been very clearly specified that there'll be no patent sought. So I don't think there's any reason why those would be encumbered, and we should be able to share with those relatively easily.
Awesome, that's great. I mean,
what organism are these being targeted against?
Trypanosoma cruzi and Leishmania. Various Leishmania, so we tested against Leishmania and Phantom. I mean, we were able to get compounds that were really pretty potent against Trypanosoma cruzi and had decent pharmacokinetics, and then they didn't work in vivo. So now we're just looking at it for leash, which is a little trickier. It's difficult to get the potency down in leash sufficiently, and we've not been able to get a compound that has any kind of exposure and still has activity against leash.
For the 814 type compounds, did you ever try putting on the aromatic rings using metal catalyzed couplings?
At the 2 position? Yeah. I can go back and check, but I don't think so, just because of how easy it is to put them in as benzaldehydes.
Yeah.
I mean, we've been working on this off and on for about 3 or 4 years, so yeah, it doesn't ring a bell, but I can go and check.
Okay.
It's a weird one, right? Just my gut feel there is that is not a particularly amenable coupling position. And I don't know, I mean, that's like completely gut feel, right? It's one of those ones where you look at it and go, ooh. Whereas at the 3 position, you can, I mean, so putting in the bit in the northwest, you can brominate that position relatively easily. If it's unsubstituted, you can brominate that and then Suzuki that to your heart's content, I think. But let me go and check.
Yeah. I think this is really quite exciting because I mean, I know everybody's concerned about the pyridine and things in terms of metabolism, but that saturented 5-membered ring also looks like a metabolic hotspot.
Oh yeah, absolutely. But I mean, you know, I think at first glance that's where one would focus the efforts, right? But I'm just in our experience, weirdly, that two pyridyl seemed to be the point at which, you know, when we finally changed that was when all of our worries disappeared in terms of mouse microsome stability, human microsome stability.
Yeah.
And we had, unsubstituted or methylated versions of the core. So 814, like where it's either unsubstituted or just methyl groups on that 6-membered ring. Which again, you'd think that's a metabolic hotspot, but when you swap the 2 position out for an NH containing 5-membered ring, imidazole, pyrazole, the metabolic stability was right down, and we got good PK.
Did you ever make the pyrimidine and look at metabolism?
Pyrimidine at the 2 position.
Yeah.
We made a bunch of, well, not pyrimidine, pyridazine, definitely. It lost a bit of potency. So whether we generated metabolic stability data at that point, I don't know. Because right now we get METSTEB on all compounds that are made, because AZ are, gifting that to Northeastern. But when Northeastern weren't making the compound, which is about half of the data set, if not more, it wasn't. We weren't getting metabolic stability data on everything. We were only getting it on potent compounds. So I have to go and check. Sorry, I'm on my phone, so I don't have the full data set in front of me.
Yeah.
And as I said, I have a manuscript that is finalized, and I'm just waiting for approval from various partners to publish it. So that'll be in CAM archive soon for about half the series. That might answer some of the questions. And there's full experimental. There's a 200-page experimental in that.
I guess a target becomes this 814 compound with the benzothiophene.
Absolutely, yeah.
To check that. So yeah, Ed and Dana, that I think is going to be an important one.
And I think, again, I'll have to go back and ask the guys in Wuhan about how they did this, but I think you can transform 814 to 818 just by doing a selective hydrogenation.
Okay.
If we did that, then we've got those conditions, and I can show them.
I was gonna say probably just NaBH4 or maybe lithium, aluminum.
I think it was a palladium or platinum, but again, it's a while ago. It wasn't active, so we never followed it up. For us,
for a representative procedure, though, Ben, can we just ping Laurie?
Yeah.
Is that all right?
Yeah, or I'll send it on to you. We worked on this in Open Synthesis Network for a while, with some people at Imperial. We had a master student at Imperial who made a bunch of compounds on this. So yeah, I've got experimentals. I just need to get my laptop and then I can see and make sure that I can upload them.
Cool. Great, that's really good. Thank you.
Yeah, I mean, making compounds exactly like the ones that you have, so the 868 type, where it's aryl at the 2 and aryl at the 3, with the saturated 5-membered. Yeah, I don't have any suggestions on that because, as I said, everything comes to this 3-component, this three reagent MCR gives you the 5-6. To get a 5-5, you don't get it.
Right.
So I don't have much I can input on that, I'm afraid.
Yeah. So
is that helpful?
Yeah, they're very helpful.
Good.
This compound that's changed it really because that's inactive. That guy. So that's one of the
that's that one I did pick up on, and that's when I went back and tried to dig out the previous. Okay. All right.
Thanks.
And the other thing I would definitely suggest on this, if you can, is trying to get an SP3 center in here somewhere, either at the 3, ideally at the 3 position. I mean, you've got some in the ring system, so it's not too bad, but just it's a bit flat at the moment, isn't it? It'll be interesting to see. I joined just as you were talking about the N-oxide. I think as a byproduct. Yeah. But obviously that's going to twist the plane a little bit, and so you're going to end up with that could be an interesting compound to check. And also, if that should tell you a little bit about the metal chelation. If that's active, then those things don't collate.
Yeah. I guess the deplanarizing also lends credit to the idea I had, which is chop off that benzothiazepine, put a morpholine on, because we have the brominated core. We just couple it to morpholine SN2 conditions. And it probably won't be active.
Yeah, that worked. It's not super straightforward, but I have conditions that we eventually worked out that did that at that position from a bromo or a chloro. I can't remember if it was palladium catalyzed or not. It was either a Buchfeld coupling or it was just a straight SNAR.
Just a quick question. Are these compounds planar, or do you get effectively atropisomers because they're twisted?
These versions, I don't know. I haven't. In ours, we have the NH in between the ring, the two at the 3 position, and so you kind of have an additional hydrogen bond that is going to maybe make it planar.
Modeling, they're very flat, but if we actually had crystal structures, that would be the ticket.
I'm sure that propeller twisted. But.
Yeah.
I mean, of course, with the way that they bind to their active site might not necessarily be the way that they crystallize, of course, right? But,
that's right. Yeah.
Fantastic. Thank you. Great. So we'll follow up with various things.
If anything else crops up or comes to mind, I will let you know. But like I said in my message, the first time I saw these was today when they popped up on a Twitter feed, and I was like, oh, okay. So that's why I dived in and said that we might be able to do something.
Makes Twitter worthwhile, doesn't it?
well, because I know it's good for being distracted, for getting depressed, and now obviously for helping out with science. So that's good.
Yeah.
And because the open source antibiotics, it's of all the OS projects that Matt is involved with, it's the one that I have the least exposure to. So I jump on it every now and then. So that's why I hadn't followed up on this. Okay.
It's all chemistry there, isn't it?
Yeah, chemistry-wise, these are nice molecules, right? And it's cool that you're able to make these 5-5s, because 5-5s, 5-5 rings are cool. And it would be, I don't know what the synthesis is of making that ring. You're making it from if I remember correctly, it was how are you making those? It's a condensation, right? You're condensing onto acetyl bromides?
Yeah.
So what's the availability of substituted versions of those pyrrolidines? The pyrrolidines that you use as the other side of the coupling part, can you get substituents on the 3 saturated carbons of that ring somehow?
I don't believe so.
That's a shame. But
which is another reason why the RR ring would be attractive.
It opens up another vector. It opens up synthetically opens up vectors.
Yeah.
Magic.
Okay.
Really
and actually, I'll have to go and check. I'm not promising anything, but I think, now I'm just thinking about it, the one thing we might have is where you have that 5-membered saturated ring. I think we might have the 6-membered ring saturated with an oxygen in it. So kind of like the morpholine imidazole as a fused core, which is a really cool molecule. So yeah, basically 818, but with an oxygen on the far right carbon instead.
Oh, nice.
It was inactive for us, but yeah. I think we've got that thinking about it. It was one of those ones that we were like, oh, that would be a cool molecule to make.
That'd be good.
Again, let me check.
Okay.
I'm trying not to promise stuff that I can't follow up on here.
Okay, thanks, Ben. Anything else? Briefly, I think we've got our hands full now with some synthesis that he's doing and checking. And we're waiting on MOA and metabolic stuff. So I think we're in a good place. We just got to make a few key molecules here, particularly now that we know that this guy is inactive.
So, Matt, I'm just thinking about you were commenting the 3 and 4 pyridyl are inactive, but they have that dioxylane at the position. So how do you interpret that?
It changes everything, doesn't it? And the assumption always is that the thing in the box is active, no?
Yeah, so you need to make those 3 and 4 pyridyl with the benzthiophene there.
Yeah,
I agree.
I mean,
just add them to the list.
I mean, it's interesting. It's a data point we needed and is interesting. It's a bit of a pisser, has to be said. Because yes, we have to reevaluate some of these SAR points.
Yeah,
it's all fun until your derivative, your parent compound's inactive.
We need to switch the one in the box to 830, right, or 822 as the generic active.
Yeah,
that's what we need to do. We need to modify this diagram.
Yep.
Yeah. Okay. So Ed and Dana, it's on the list. 3 and 4 has to be. Has to be. Okay, really good. Thank you. That's very, very helpful. Wow, really good ideas. Thanks for coming. We'll try and have another meeting next week as per usual.
Okay, that's great.
Till then, stay safe.
Okay. Bye then.
And, see you soon.
Bye. See you.
Bye.
all right.